Yeah, so I work with John Cook at the Department of Biology and the Berkeley Museum of Natural History. I'm going to be talking today about a project I started all of 6 weeks ago with some collaborators down at the California Academy of Sciences, using a new library preparation method known as hyRAD to investigate the phylogeography of a New Guinea forest kingfisher. And my interest in this project began because my nascent dissertation work looks at the factors driving divergence between this lowland species, which is the yellow-billed kingfisher, or Cynotora, and its sister species, the mountain kingfisher, Cynomegorhyncha. And there's a lot that's really cool about these birds that I'm going to have to skip over for now. But because I'm broadly interested in species limits and the role of natural selection in driving their divergence, I first needed to determine the phylogeographic patterns in the lowland species in order to have a robust sampling scheme for later stages of the project. But that's actually a really interesting question in and of itself, because the island of New Guinea, if you're not familiar, is ringed with what appears to be, at least from above, contiguous tropical lowland rainforest. But avian phylogeographic studies don't show that pattern. So for instance, this is a figure from a paper by Diner et al. In 2009 into the phylogeography of the bird known as the little shrike thrush, which had, or has at one point, been classified with 24 distinct, 24 named subspecies. And what's more, some of these subspecific breaks concord with major biogeographic breaks that have been hypothesized to have resulted from incursions of the ocean during the Pleistocene. So while we have some information on species like the little shrike thrush, we don't know how widely these patterns are shared across taxa. Compounding this problem is the fact that in order to do the sort of dense, geographically widespread sampling you need to answer a lot of the questions you're interested in, well, it's really difficult in New Guinea because it's politically tumultuous and exceedingly remote. However, there's a great resource in the form of historical collections, from expeditions like the American Museum of Natural History's Whitney South Seas expedition, Bravo Vector, he was on it, that are a really great resource. But of course, those specimens were not collected with modern tissue samples available these days. So that introduces the problem of how to do genome-wide population genetics or phylogeographic work with highly degraded and low copy number DNA that's typical of data extracted from museum specimens. And I'm going to refer to that as ancient DNA. That's a bit of a misnomer. So you're no doubt familiar that next generation sequencing methods are ubiquitous now and work with non-model organisms. But it's been harder to get them to work with ancient DNA. So for instance, a method like sequence capture, UCs, exon capture, in phylogenomic studies can be used with modern tissue or ancient DNA. For population genomic work, it works well with modern DNA or ancient DNA if you have a transcriptome to find probes from. Similarly, RAD-seq, wildly popular for population genomics with modern DNA, but there's still kind of a gap for doing population genomic work if you have degraded DNA, but you don't have a transcriptome to design probes from. So one possible solution to this problem was proposed in a paper published earlier this year by Thomas Suchan and colleagues in PLOS One, and that's a method known as hyRAD. And hyRAD is more or less what the portmanteau suggests it is. It's got a mix of sequence capture or hybridization with restriction-associated digest DNA sequencing. And the basic idea of it is you take, you do a typical RAD digest, and then take those libraries and turn them into biotinylated probes, which you can then use to theoretically hybridize with orthologous loci in your other samples of interest. You can enrich those regions for sequencing, and it shouldn't matter whether the loci you're capturing or the sequences you're capturing are degraded or not. So this seems like a really good option for working on phylogeography in a place like Guinea. So our initial goals, in addition to investigating the phylogeographic patterns in the yellow-billed kingfisher, were more or less just to figure out whether we could make hyRAD work efficiently, since it's such a new method, and weren't entirely confident it would work first pass through. And there's a lot of room for customization and tinkering in the published protocol. Well, I tried to kind of hustle everything over the finish line. It's mostly the second point that I'm going to be talking about. So our initial sampling of the yellow-billed kingfisher attempted to represent all named subspecies and the full range of the species, with the exception of a tiny population, or a small region in the Cape York Peninsula of Australia, where the yellow-billed kingfisher is also present. And of our samples, 14 were historic or ancient DNA extracted from the topads and museum specimens, mostly clustered in West Papua, which is where there's less modern ornithological work being done. And then we had 7 samples that were from modern tissue collections. And so we were able to take all 21 samples, perform pool hybridization reactions, 3 pools, and they all successfully amplified, and we've shipped them all off to the HiSeq. But we were also able to run the 7 modern samples on MySeq and based in the Cal Academy. So we have some preliminary data on how well that actually works. But first, a quick note on the Bioinformatics Pipeline. Tomas and his colleagues did present a custom Bioinformatics Pipeline for dealing with hyRAD data, but for whatever reason we kind of forged that on our own. And using a modified version of an exon capture pipeline put together by KB at Berkeley, we combined that with hyRAD to produce alignments to analyze. So the basic idea is we first took the RAD library, which is our probe library, cleaned and filtered it, clustered some of those reasonable loci, and then we blasted that against itself in order to drop duplicates, drop repeat regions. We then went to the hybridized library, capture reactions, cleaned and filtered those, clustered those, and then aligned those with our reduced probe library, which was sort of a reference genome, since we don't have a genome for Cynotora or indeed any kingfisher that I'm aware of. We then, after using that and exporting that for phylogenetic analyses, merged all our assemblies together again to make sort of a master assembly of the probe regions and then the flanking regions, and then aligned raw reads back to that to get coverage. So one of the things we were really curious about with our initial MySeq runs is just how evenly the reads were going to be distributed across our samples, especially since we were pulling sort of high molecular weight DNA from the RAD library with our reduced capture libraries. And it was nice to see that there's actually not a ton of variation. It goes from about 6% to 22% of the reads total, but most are right along the middle there. A more significant worry was whether or not we were actually going to be able to capture regions of interest, the on-target regions from our probe sequence. And so what this figure shows is sort of our stepwise loss of the total number of loci as we proceeded through the analysis. So for instance, after assembling our probe library, we had north of 500,000 loci. After removing repeat regions, that dropped to around 160,000 loci. Likewise, with our hybridization reactions, started with between around 14 to 22,000 clusters, which then was winnowed down to 4,000 to 9,000 loci that were actually on target with our probe regions. And we don't know how many of those are shared among our different samples yet, but that's an obvious next step to our study. And because I really wanted to show something that wasn't just a bar plot at this talk, we also worked to take our off-target reads and assemble mitogenomes for the 7 captured samples. And you probably are aware you can do this, and it's a pretty cool use, and so much of your Lumen run is usually this discarded. And we basically took our raw, or I shouldn't say rad, but our raw fast keys from the MySeq read, cleaned and trimmed those, and blasted them against a previously published mitogenome from a really close relative of Simon. We then took this to a de novo assembler known as PRICE, which stands for that paired read iterative contig standard. And what that does is it kind of takes your paired end reads that have a little bit of overlap with existing contigs, and then works out iteratively in cycles until things start asymptoting. And with this, we were able to get mostly complete mitochondrial genomes for our 7 samples. They looked pretty good, just a really quick eye check of most of them. Looked pretty good, except for the control region was super dodgy. So we dropped those when we aligned them. And I just learned the RAXML, so both to, again, have a figure that we can bar plot, but also in order to just check basically for contamination or other things that I'd be worried about later on in the process. And the important takeaway here is that even though there's low support deeper in the tree, I think because S. tortorocratia there had the sort of patchy assembly, the previously named subspecies are clustering together, and they sort of line up with our a priori expectations of biogeographic barriers, which are the acronyms there in green. So in summary, especially since we were able to successfully amplify the reactions from the ancient samples as well as our modern samples, we think this is going to be fairly robust for our use going forward. But we definitely came across some downsides. Most notably, had a lot of problems losing DNA throughout the process, both in de-adapterization, which we can't really explain, and then especially during biotimolation, because the method we were using involves an enzyme that's bionic, and it kind of chews up your fragments. So because of this, we had to amplify our probe library quite a bit, which meant that we had some PCR jackpotting, and it just will probably take more coverage overall in order to have relatively even coverage across loci. So my colleagues are working sort of a modified version of the protocol that should address some of these concerns. So our next steps, I'm really looking forward to getting the HiSeq run back in a week or two. And after running that through the analysis and kind of vibing on that for a while, we're either going to go back and fill in some of the gaps in our sampling scheme with Simon Toro, or depending on that, potentially go on to sample the highland species, Simon Megalinka. So on that note, I'd like to acknowledge my supporting institutions, sources of funding, and especially my collaborators, Jack Dunbacher, Anna Sellis, Zach Hanna, and then my advisor, John Plickett. I think I have a little time for questions. Yeah.
Do you have mitogenomics from all the samples you captured?
Yeah.